This is an A-level biology presentation looking at the regulation of transcription and translation. In this presentation, we're going to look at transcription factors. We're going to look at the role of transcription factors, and we're going to specifically look at the transcription factor estrogen and its ability to regulate transcription. As well, you're going to have to go and do a number of questions because most likely in the exam they will ask you to interpret data from investigations into gene expression. So they'll ask you to apply the information that you know about this particular topic to interpret data from some sort of research. And the best way to do that is get hold of as many past paper questions from textbooks, online from different websites, or from the website of the particular exam board. They'll have past papers. To get started, you should know the 4 types of stem cells. You should know the difference between those types of stem cells in terms of a basic description, and you should be able to say where those particular types of stem cells are found. So you should be able to complete the table in front of you. Pause the presentation, and whenever you're ready, go on through your answers to the best of your ability. You can restart and check your answers. So pause now. In terms of stem cells, there are 4 types of stem cell: totipotent, pluripotent, multipotent, and unipotent. In terms of description, remember stem cells are split up based on their ability to the number of different types of cells they can differentiate into. So totipotent can differentiate into the most and widest range of cells, whereas unipotent can only differentiate into one particular type of cell. In terms of where they're found, again, totipotent are found in zygotes, which is why they're less and less used in research because of the ethical issues. Pluripotent are the most common ones, and they're found in things like fetus and embryonic stem cells. And then we can start going into multipotent from umbilical cords, and adult stem cells can be multipotent. And again, the unipotent are normally only adult stem cells. Before you go any further through this presentation, you have to be 100% secure in your knowledge of the processes of transcription and translation. Remember, transcription is the process of getting a DNA strand and using it to produce an RNA strand, whereas translation is getting an RNA strand and using that to produce a protein or polypeptide. At the bottom of your screen, there are a set of pictures that you need to be able to link to the processes of transcription and translation. You need to be able to use the terminology helicase, complementary, hydrogen bonding. You need to be able to talk about a template strand, codons, anticodons. You need to be able to talk about the P site and the A site, the large subunit of the ribosome, the small subunit of the ribosome, start codons, end codons. There's a whole raft of terminologies you've got to be 100% secure on. If you are not, please go back and look at the presentations and go through the transcription and translation presentations, because if you go any further, you're going to find yourself getting very, very quickly confused. So how do we actually stop particular polypeptides or proteins from occurring? Well, we actually turn the entire process off at the start in terms of transcription, because if transcription cannot occur, we don't produce any messenger RNA. If we don't produce any messenger RNA, then we can't have translation to produce any type of polypeptide. In terms of turning it off, transcription only occurs when you have a transcription factor bind to the DNA as a starting point, and that is where we switch genes off by stopping that binding to a particular promoter site. If we don't bind to the promoter site, transcription does not occur and the whole process stops, and we've switched the gene off. If a gene is switched on, it means that the helicase can act on that particular section of DNA to unzip that particular DNA to show a template strand. We will have a transcription factor, we will have a promoter site, we will have binding, we will have the entire process of transcription occur. Remember, this is talking about pre-transcription. We're talking about the start of transcription, the turning the ignition on in the actual engine as opposed to how the engine works. In terms of transcription factors, a transcription factor doesn't randomly just drop on DNA in the hope of finding the right place. What transcription factors do is they just bind any section of DNA they can get into, and then they move along that particular strand of DNA until they find a specific base sequence, which is the complementary binding site for that particular transcription factor. Once that particular transcription factor finds its binding site, it'll obviously bind to the DNA. And when we say it binds to the DNA, the structure of the transcription factor changes. With a change in the transcription factor, other proteins are able to interact and bind on, and essentially we have the start of the entire transcription process. If we cover up the binding site, if we stop the binding site from being exposed, if we stop an interaction between the transcription factor and the binding site, then transcription will not occur. There is no messenger RNA, and as a consequence, we cannot have translation, which means we have no polypeptide. Remember, if you get questions on this on the A-level exam, you must follow through the entire process. You can't just stop with, oh, the transcription factor doesn't bind. If it's a 2, 3, 4 mark question, you've got to lead all the way through the process in terms of how it stops the production of messenger RNA and how that stops the production of polypeptides, and as a consequence proteins are not produced and certain processes don't occur, enzyme reactions don't occur, or certain characteristics and pigments aren't produced. You've got to follow it all the way to the end, particularly if it's worth more than 1 mark. In the AQA specification, you are specifically asked to go over estrogen as a transcription factor. Use the images in front of you to try and work out this explanation of how estrogen acts as a transcription factor. When you've made your best attempt at it, you can of course go through the next bit of the presentation and see how you've got on. So pause the presentation now. The first thing you have to explain on the diagram is where the estrogen actually comes from. And the estrogens come from outside of the cell. So you need to talk about the fact that the estrogen moves across the cell surface membrane and it moves by diffusion. The reason we know it's diffusion is because in the diagram of the cell surface membrane there are no proteins present. If it's moving by diffusion, that means that the estrogen must be lipid soluble. Once the estrogen is in the cytoplasm, what it can then do is bind to a complementary receptor on a DNA binding site. And the DNA binding site is part of the transcription factor. When the estrogen has attached, the estrogen then causes a shape change in the DNA binding site. Because we've had a change of shape in the DNA binding site and the whole transcription factor can now move in through the nuclear pores and into the nucleus. Once it's in the nucleus, it can bind with the DNA strand, and as a consequence, it can start the process of transcription. What you've been given is a number of terminologies, some of which link to this presentation, some of them are linked to other presentations. You have to know all of them as part of the A2 course for AQA, so please take your time to make sure that you have memorized, and if necessary go back and look up the presentations and or definitions and learn them off by heart. You can pause the presentation now, go through your definitions, and when you're ready restart and check your answers. So these are the definitions for each of the terminologies. Again, make sure that you're not just able to repeat the words on screen, you have to actually be able to explain to someone what that concept actually means as opposed to just regurgitate, because they will bring these in to explain questions. Don't be afraid to go back over other presentations, or again just use textbooks, notes or other sources of material to go through and make sure these concepts are in your head.